All right, so we are continuing with the auto ionization of water. So in the last video, we said that for neutral water, the concentration of this is equal to 1.0 times 10 to the negative 7. And the concentration of this, the molar concentration of this is 1.0 times 10 7. So this is M, this is M, indicating molar concentration. Now, usually when you're reporting this, sometimes you don't want to use this number because it's cumbersome. So to avoid that, what people did was to report this concentration in terms of logarithms. So these concentrations can be reported in terms of logarithms because they are cumbersome. So we want something that we can easily deal with. So what we are going to say is I will take the logarithm of this concentration, which will be equal to the logarithm of this concentration. Now, if I put this concentration in logarithm, it will be 7. Now, instead, it will be minus 7. So we say, no, we don't want to have minus. So we don't want to have minus. So what did they do? Instead of taking logarithm, they took minus. If we take minus logarithm of the concentration of this, will be equal to minus logarithm of the concentration of this. This will give us 7. So what they did is they defined something. Definition: pH. pH is defined as minus logarithm of the concentration of the hydronium ion. So that's how pH is defined. So it is something that we are going to look at in the next slide. We also define pOH. pOH is defined as minus logarithm the concentration of OH minus. So these are the two definitions that we are saying. pH can be defined as negative log H plus or negative log H3O plus. We know that H plus is equivalent to H3O plus. They are basically the same thing. While pOH will be defined as negative log OH minus. Now something else. Kw, we said that Kw is equal to 1.0 times 10 to the power negative 14. That's Kw. Now, if you take negative log Kw, this will be equal to negative log 1.0 times 10 to the power negative 14. This will be equal to 14. So what we are saying is this is the 14 that we have here. Why do we have 14 there? Because it has come from H3O plus or H minus is equal to 1 times 10 to the power negative 14. Now, if you take the logarithm of both, you do the same thing here, log of this. It will give you 14. That's the 14 we have here. So let me just create some space by getting rid of this. So we say pH is negative log H plus, POH is negative log OH minus. But if you add pH plus POH, they are going to give you 14. Normally, that's the reason where the pH scale ends with 14, because we are using water. So if you want to measure pH and using water as your standard of measurement, the pH scale will go to 14. So let's take note of that. We are going to use, this will be equation 1, this will be equation 2, this will be equation 3. We are going to use these 3 equations to determine pH of solutions. First question, what is the pH of a 0.25 molar of HCl? So in our solving, we said pH is negative log of H plus. So what you are saying is that HCl here, the concentration of HCl is equal to 0.25 molar. This is the same concentration of H plus. So the concentration of HCl is the same concentration of H plus, because this is the H we are talking about. So this one is negative log 0.25, which is log 0.25, which is 0.60. So the pH of this solution is 0.60. So it's pretty acidic. Now, what is the pH of this 0.082 molar of sodium hydroxide? So to solve this one, first determine the POH. Now we know that pH plus POH is equal to 14, because we can easily calculate pOH and then determine pH from the pOH. Now, pOH is equal to negative log the concentration of OH. So here, we know that the concentration of sodium hydroxide is 0.82 molar. This is the same as the concentration of OH. Again, 0.82 molar. So we are saying this one will be equal to negative log 0.82. So negative log 0.82. So it gives us 0.09. That is the pOH. But we are saying pH plus pOH is equal to 14. In other words, we say pH plus 0.09 is equal to 14. So we can say pH is 14 minus 0.09, which is 13.9. So it is really basic. It's really basic solution. The first one here is really acidic. The second one here is really basic. But that's how you calculate the pH. If you are given acid or if you are given base. Here, you are given acid, you just plug in the acid into the pH. Here, you are given a base, you need to calculate the pOH first before calculating the pH. Now let's talk about buffers. I introduced buffers in the previous topic. Here we'll look into them more than we did last time. So buffer solutions will resist change to pH. Now, if you have a buffer, if there is an acid added to the buffer is going to neutralize the acid. If you add a base to the buffer is going to neutralize the base. Now, these buffer solutions are usually mixtures of weak acid. So we need to be focused here. You need to have weak acid and conjugate base, its conjugate base. So we are saying you can have this weak acid and its conjugate base. So this is a buffer solution. Or you may have a weak acid and its conjugate base. If you have a strong acid, so strong acid and strong bases don't make buffers. So strong acids and strong bases don't make buffer solutions. So you are not going to have a buffer solution if you have a strong acid or strong base. So really an example of a buffer reaction If I can have CH3, COO, H, aqueous, plus H2O, liquid, I get CH3, COO, negative, aqueous, plus H3O plus, aqueous. So if I have a buffer like this one, if I add acid, acid is neutralized by the base CH3COO minus. If I add base, the base is neutralized by acid CH3COOH. Actually, I need to put double arrow here. All the buffers will have double arrows because they are weak acids or weak bases. So this is what happens. If you add acid, it is neutralized by the base in the buffer. If you add base, it is neutralized by the acid in the buffer. So you need to be able to tell me that. I will ask you what happens if I add acid, what happens if I add base to a buffer solution. So some examples. Usually, actually, buffers in the body and in the lab are very, very important. If you are in the lab, you want your buffer. Usually, we use the buffer that I showed you before. It is known as acetic acid. So this is acetic acid, and this is acetate. So it is called acetic acid acetate buffer. We use it a lot in the lab to buffer our solutions. Then the buffer that is used actually mostly in the respiration, actually this should be H2, not H3. It is carbonic acid, so this is carbonic acid, and this is hydrogen carbonate. So it's carbonic acid, hydrogen carbonate buffer. This one is used in the body like extracellular fluid. Like in the blood, so you have this buffer to deal with the pH changes. Then we have phosphate buffer. So this is phosphate buffer. This is phosphoric acid. This is its conjugate base, phosphoric acid and dihydrogen phosphate. So I'll say dihydrogen phosphate. So this is also used, now this is intracellular fluid inside the cell. So fluid inside cells. This is outside cells. So these buffers are the ones that maintain the body, the physiological pH of the body. Then we have hemoglobin buffer. Hemoglobin buffer will have hydrogen attached or oxygen attached. So this is hydrogen attached to the buffer or oxygen attached to the buffer based on the pH. So all these buffers work, especially this one, this one, this one. They work to maintain human fluid physiological pH of between, I'll just say, of 7.4. So pH is equal to 7.4. That is the physiological pH that all these buffers are going to maintain in the body. So when we say buffer solutions, an important thing is they are made of weak acids and weak bases. Like if you look at this one, HCl, Cl, this is not a buffer because HCl is a strong acid. The same thing with HNO3. The same case, not a buffer, because HNO3 is a strong acid. The same with H, we say NaOH, H3, H2O, not a buffer because NaOH is a strong base. Now, if you have strong bases and strong acids, you are not going to make a buffer solution. So for now, let's stop here. Thank you.